right, so talking about immunity, and although we don't quite get to it in this slide, do single-celled organisms have immunity? So do they have an immune system? No, because they have no body systems really. But they do have protection against pathogens. So there are viruses that infect even tiny little bacteria. Tiny little bacteria can be infected by a virus. And so even single-celled organisms have enzymes, for example, that can destroy pathogens that have entered the cell. So there are immune responses in even simple organisms. We're going to talk more about the vertebrate immune system, talk specifically about humans. And so the human immune system consists of mainly white blood cells and proteins. We'll talk about some of the specifics. Okay, so white blood cells, technically they're called leukocytes, are a part of our immune system. And they are nucleated cells. So our red blood cells are non-nucleated. There's no nucleus in a red blood cell. The white blood cells are nucleated. In fact, some of them have more than one nucleus. Typically, they are less numerous than red blood cells. If you were to look at a blood smear or do a blood count. Some are larger than red blood cells, some are similar in size. And there's actually several types of white blood cells. The immune response to an infection is really complex. And there's several parts and several types of cells, and some cells produce other cells, some produce antibodies. So it's a pretty advanced system that has a lot of parts to it. We're just going to sort of talk about the basics. So one type of immune cell, one type of leukocyte, is called phagocytes. And the two types are monocytes and neutrophils. What does this word look like? A word you've heard before. Phagocytes. Dean?
Phagocytosis.
Yeah, what is that, phagocytosis?
It's like with the pseudopods. What's the word?
Engulf?
Yeah, engulf something.
Yeah, phagocytosis is when a cell engulfs something. And so phagocytes are blood cells, white blood cells, leukocytes, that do that. But what they ingest and destroy are pathogens, bacteria, viruses. So phagocytes can engulf and destroy these pathogens. They are circulated throughout the body in the blood, but they can exit the blood vessels to a site of infection, okay, and destroy those pathogenic cells at the site of infection. And when we do get an active infection, our immune system ramps up the production of these cells so that they can go and hopefully destroy the pathogenic cells and help us to get better. So these are the other types of white blood cells, the types of leukocytes. We're not going to really get into all the details of how they all work. So lymphocytes are another type. We see them here. And lymphocytes are cells that come in different varieties. Major categories are B cells and T cells. And what they do is they are the cells that can produce antibodies. And we'll talk about antibodies in a second. So antibodies are protective proteins. These lymphocytes give us our long-term immunity. So the phagocytes destroy an infection that's currently active in the living thing. Okay? And these lymphocytes help to establish long-term protection against those same pathogens. They're produced in the bone marrow. We have an abundance of lymphocytes in our lymph tissues. So in our tonsils, adenoids, in our lymph nodes, there are many of these lymphocytes that help to coordinate our immune response. And so they produce antibodies, which are, like I said, are proteins. And what's always, whenever we're talking about proteins, what's important about the protein? Just talked about this the other day. We talked about hormones. The shape. And it's also important in antibodies. The shape of the antibody is crucial because the shape of the antibody matches with the shape of proteins that are on the pathogen. They're called antigens. Okay. So let's say this is a chickenpox virus and this is a measles virus. They have on the outside, just like our own body cells have antigens to say, hey, I'm one of you, don't kill me, to our immune system. Well, pathogens also have these sort of identifying proteins on the outside, and these are the proteins that our immune system sort of looks at to see if they're recognized. So when our body produces antibodies, when our immune system produces these antibodies, the shape of those antibodies matches with the shape of the antigens. And so they're specific. An antibody is specific to one pathogen. So the binding sites are shaped differently because each of these antibodies interacts with a different antigen and a different pathogen. So what happens is when we have produced antibodies, they're floating around in our circulatory system. If they encounter the pathogen that they match with, they start to bind to it. The antibody binds to the antigen on the surface of that pathogen, and they start to clump up. Okay? The antibodies can also cause changes inside of that pathogen that cause it to burst, destroying it. They also mark it so that macrophages, other white blood cells, okay, other phagocytes can come and destroy and engulf it. So these antibodies do several things. Okay? They prevent it from infecting cells, they clump them together, okay, and then mark them so that the phagocytes can come and actually destroy them. So where do we get these antibodies? Well, they are part of what we call active immunity. So our body produces these antibodies when exposed to pathogens. And they can last a long time. Some of these antibodies last for our entire lifetime. Others are more temporary. So sometimes we get this natural immunity. For example, I got chickenpox when I was a kid. Okay? So when I was a kid, everyone got chickenpox eventually. Chickenpox is caused by a virus. Okay? You catch it from other people. Makes you sick. Gives you these bumps on your skin that are really itchy. And so I got chickenpox. I was sick for a few days. My immune system kicked in. Okay? Fought off the infection. My lymphocytes produced antibodies now that match with the chickenpox virus. And they're still in my body today, these antibodies. If someone were to get chickenpox and I got that into my system, the pathogen, these antibodies would quickly bind to it, and then phagocytes would destroy it, and I would never get sick. Because my body has these antibodies that get the immune system response to it happening very quickly. And so you generally, a person doesn't get the chickenpox twice. Because you have antibodies once you get it one time. So that's natural immunity, getting sick. And people have noticed this for thousands of years. Even in ancient Greece, people noticed that if a person got sick with a certain type of plague, but recovered from it, they wouldn't get that same type of plague again. They were protected in some way. Eventually, people learned about how to create vaccines. So vaccines are an example of acquired immunity. One of the first vaccines, the first vaccine actually, was made by a guy who had a hunch that it would work. And so he took cowpox fluid and he put it into an 8-year-old boy, thinking it would help protect them from smallpox, which is related, which is deadly human disease. Kind of risky, but it actually worked. It's one of the first types of vaccines. Scientists soon learned how to do this properly without just injecting pathogens into somebody. And so that's an example of acquired immunity. So you've all been vaccinated likely for a variety of things. For example, my daughter, so when my oldest daughter was born, the chickenpox vaccine was just starting to become used. Okay? Before that time, people got the chickenpox. After, so my daughter got the chickenpox vaccine. When you get a vaccine, how many people got a flu shot this year? Yeah, usually about half. Okay. Flu shot is another vaccine. So what are they actually injecting into you? So when you get a vaccine, you go, you get a shot. Brady, what are they doing?
I don't think it'd be part of the actual disease, so your body goes undefined enough that it actually gets it.
Yeah. So are they injecting the flu virus into you?
It's like a week.
Zach, could you say that? What?
It's weakened or dead.
So yeah, so this is how this is, and thinking ahead to the Regents exam or your test next week. This is a common question on the Regents exam. What's in a vaccine? How does it protect you? And so forth. Questions like that. And one of the reasons students often get this wrong is they say a vaccine, they inject a small amount of the virus into you. Well, that's not true. Because what if they injected a small amount of the flu virus into you? You get the flu. So they're not injecting a small amount, but they're injecting a weakened, sometimes it's a weakened form of the virus. Sometimes it's just a part of the virus, not the actual entire thing, or it could be a bacteria. So there's different types of vaccines that can be administered, but it's never the active, live pathogen. Okay, that would be a bad idea. So when they inject the flu vaccine, yes, it is an inactivated, weakened, dead version of influenza. Even though it's weakened and dead, your immune system kicks in once you get that injection, and your lymphocytes will start to produce antibodies against that pathogen. So if, as the year goes on, if you're exposed to that same version of influenza, you already have the antibodies. So your antibodies kick in, they ramp up your immune response very quickly, and you fight it off quickly without actually getting sick. That's how all of these vaccines work. Okay? By injecting some weakened or dead or part of the pathogen into you, your immune system is primed to produce antibodies which protect you for a long time. Some last for basically your whole lifetime. Others you need to be sort of updated, like tetanus. You have to go for a tetanus booster, they call it, every so many years, okay, to maintain that immune response, maintain those antibodies. Now, vaccines aren't perfect. Like, you know, I'm sure that you can get a flu shot, a flu vaccine, but still get the flu at that point in the year. That's because many types of pathogens are constantly mutating and changing. And if they mutate in a way that the antibodies no longer recognize them, then it's not protective. Sometimes there's lots of different strains or versions of the flu that make their way through a population. And if the vaccine that was produced, they try to predict what are going to be the most common forms of the flu that are going to go around next year, because they have to do this in advance. If they sort of don't predict correctly, and the strain that's going around is a different strain, well, it might not be as protective. So that's how vaccines work. Vaccines are one of the primary reasons for expanded quality of life and lifespan throughout the 20th century. The development of vaccines was huge in terms of human health. this is polio, for example, debilitating, handicapping disease that affected tens of thousands of people every single year. Okay. So you see the levels of polio rose and fall throughout the years, peaking up here. The polio vaccine was developed and started to be used in a widespread manner in 1955. That year there were 29 or so thousand cases of polio. Look what happens to the amount of polio in the next couple of years. It just drops, and then 10 years later, basically nobody gets polio. The levels are almost zero. So this vaccine has helped save thousands and hundreds of thousands of people from this disease. So vaccines, we have each sort of several years, new vaccines are developed, vaccines against new diseases. And so there are requirements for kindergarten. To go into kindergarten, in most states, there's a requirement that a person be vaccinated against a list of diseases. And this is to help protect general public health. You can get a waiver in some states depending on that you have some objection to your child becoming vaccinated, and they can come into school without it. So you probably have heard in the news lately that there's more and more people that are deciding not to vaccinate their children. And so what's happened is there's certain diseases that have been pretty much very, very rare in the United States. They're starting to make a little bit of a comeback. You know what one of them is that's happening in little pockets around the country? Measles, yes. So most people have been vaccinated against measles, and so you get a vaccine, MMR vaccine, and you build up antibodies for the measles virus. And so if, let's say in a school situation, if everybody's vaccinated for measles, well, no problem, right? Because no one's going to be carrying the virus, so likely it's not going to be present in the population. If you have one or two people that aren't vaccinated, probably still okay, because most people are not going to be exposed to the virus, are going to still be healthy. But as the number of unvaccinated individuals in a population grows, well, now there's a little reservoir of people that are not resistant that could be carrying the virus. But the other thing, and it's not just the unvaccinated people that are more at risk. Actually, all the vaccinated people are at risk too, because the vaccine's not perfect. So let's say if all of you have been vaccinated for measles, it's not 100%. One or two of you might still have the possibility of getting the measles. But as long as just about everybody around you is vaccinated, you're not really going to be exposed to the measles virus, so you're not going to get it, even if it didn't work. But as the number of unvaccinated individuals grows, there's a reservoir for that virus to be there. So even those that are vaccinated have a higher risk of actually getting the disease. So that's sort of that's called herd immunity, that concept that the more vaccinated individuals in a population, the more protected everybody is. All right, so we're going to watch a video here, just a little bit about sort of how vaccines work. That's kind of to reinforce some of the things we just talked about. And then we have another page and then a little bit more, another video. So before our immune system is needed, we also have other means of preventing pathogens from even getting into the body in the first place. Okay, our initial protection against pathogens. And so some of those are, for example, your skin. Your skin is relatively impermeable to any pathogens, right? Typically, you're not going to get a pathogen into your body through skin as long as it's intact. Your respiratory tract is often a means of pathogens getting into your body. So you also have things like mucus in your nasal passage, okay, helps to trap these pathogens. Cilia, tiny hairs in your nose, in your trachea, help to filter out these things. Ingesting pathogens is sometimes a means of infection. Our stomach acid helps to destroy many of these pathogens. They can't survive those conditions within our stomach, so they don't take hold. tears have pathogenic qualities to them. Also, having a healthy gut microbe population is important. We talked about this when we talked about the digestive tract, that our gut microbes are hugely important to us. Right? If we have a healthy population of beneficial gut bacteria, any harmful bacteria that gets in doesn't have a place to take hold. It's outcompeted by all these healthy bacteria in our digestive tract. However, if we have compromised gut bacteria, maybe we're on long-term antibiotics, well then there's space for these pathogenic bacteria, like C. diff, to sort of take hold and start to cause an infection. So healthy gut bacteria are very important. If pathogens get through all these barriers and start an infection, that's when our immune system takes over to help fight off that pathogen and then provide future protection, hopefully. So if you remember when we talked about gut microbes and so forth, embryos and a fetus is basically as a sterile digestive tract without all of those beneficial bacteria. But within the birthing process is when these helpful bacteria are actually introduced into that newborn. And so there's some recent research about comparing gut microbiota of babies delivered via C-section versus babies born vaginally. And there's a big difference. Which version do you think has the full complement of gut microbes? Yeah, after a vaginal birth. Because the process of labor and delivery takes bacteria from the mother, and they then inoculate the baby that's being born. When C-section birth is done, that process doesn't happen. There's no vaginal birth. The baby's taken out through an incision in the abdomen, and so then those same microbes are not introduced into the baby's system. So we're going to just learn a little bit about this through this video. It's kind of interesting, some of the research that they're doing. Does anyone know anything about rates of C-section birth in the U.S., for example? Are they increasing, decreasing, same? Anyone have a guess? Not sure. Ma'am? Which one? They have increased. Sometimes when a woman is trying to choose like a hospital to give birth at, one of the things they sometimes look at are the rates of C-section, okay, because that varies from hospital to hospital, from doctor to doctor. And this video will sort of talk a little bit about that as we watch. So you can take a break.
They were born